So we have the Acinar Cells, and it's by myself, Colin Fitzpatrick, Oisin Feely, and Oisin Fitzgibbon. So just a brief introduction to the Acinar Cells and some of the things they actually do within the pancreas. So they're found in the pancreas and are key in degradation of specific biomolecules. The pancreas is made of both exocrine, so ductal, and endocrine, ductless glands. Acinar cells can make up more than 95% of the overall cellular population in the exocrine pancreas tissue. The cells are primarily involved in the synthesis of digestive proteins along with enzymes and actually have the highest level of protein synthesis within adults. They are the specialists in synthesizing, packaging, sorting, and regulated secretion of digestive enzymes and isoenzymes. They also play a vital role in pancreatic injury and disease, which we will discuss further. So basically this is just an overview of the pancreas. So the pancreas is an elongated gland situated transversely across the upper abdomen, which is located behind the stomach and the spleen. As stated in the last slide, the pancreas is both an exocrine and endocrine gland, each with their own specific functions. The exocrine tissues, which are the acinar tissues, produce important digestive enzyme precursors that get transmitted into the small intestine. The endocrine tissue, which is contained in the islets of Langerhans, produce two key hormones, insulin and glucagon. Both of these hormones regulate carbohydrate metabolism. Other hormones that are produced in the pancreas are vasoactive intestinal polypeptide and somatostatin are important in terms of intestinal secretion and motility. Excuse me. So here we just have the actual pancreas. So you can see it's not a full view of the pancreas. So you can just see the acinar cells here in the top right hand corner, and just below that you can see the capillary network, and then you can see the alpha cells, which are the kind of light green cells, the beta cells, which are the kind of purpley grey colored cells, and the delta cells, which are the brown cells. And then you can also see the islet of Langerhans, which contains all of those cells, so the alpha cell, the beta cell, and the delta cell. You can also see the central duct. So the acinar cell structure. They have the shape of a truncated pyramid arranged around a central ductal lumen. Acinar cells are larger than centroacinar cells and are easily identified because of the darkly stained zymogen granules. So zymogen granules are specialized inactive storage organelles in the acinar cells that regulate apical secretion of digestive enzymes from the cells. The basal portion of the acinar cells lies next to the interstitial space that contains vessels, nerves, and connective tissue. Nuclei and nucleoli are in the basal portion of the acinar cells. The Golgi lies at the junction of the basal and apical portions of the cell. Mitochondria are generally located around the nucleus. Centroacinar cells have less rough endoplasmic reticulum and no secretory granules. Their cytoplasm is more lightly stained. So here you can just see kind of an overview of the acinar cell structure. And you can see some of the things that we've spoken about, the zymogen granules, the Golgi, you can see the Golgi vesicles here as well. And you can see also the mitochondria that are located near or around the nucleus. You can also see clearly identifiable the nucleus, and then right at the bottom you can see the basement membrane. So the acinar cell structure. The central ducts then filter into progressively larger intercalated and collecting ducts before moving into the pancreatic duct. Once the enzymes reach the pancreatic duct, they are then secreted into the duodenum and known as pancreatic fluid. Acinar cells produce the enzymes to degrade dietary proteins, fats, and carbohydrates in the intestine. Another protein secretion is trypsin inhibitor that keeps calcium in solution, and also albumin and immunoglobulins. The enzymatic proteins are synthesized on ribosomes from amino acids carried to the pancreas by the bloodstream. So, Acinar cells and enzyme secretion. The enzymatic proteins are synthesized on ribosomes from amino acids carried to the pancreas by the bloodstream. Enzyme precursors are conjugated in the Golgi and then concentrated into membrane-wrapped zymogen granules. These are generally stored in the cytoplasm before secretion. The enzyme secretion is controlled by stimulants, for example, secretin that is released from the duodenum from the introduction of gastric acid. The CCK or acetylcholine bind to specific receptors on the acinar cell membrane. So what happens during enzyme secretion? The CCK or acetylcholine bind to specific receptors on the acinar cell membrane. The binding of the CCK to the receptors in the membrane of the acinar cell causes the production of bicarbonate and enzymes. The zymogen granules migrate to the apex of the cells where they pass into the central ductal lumen. Through the binding of vasoactive intestinal polypeptide or secretin, bicarbonate production increases along with sodium, water, and enzymes. These come from the acinar cells and small ductal cells. The bicarbonate is secreted in exchange for chloride, with sodium being replaced by hydrogen, causing an overall decrease in pH of the blood leaving the pancreas. In the absence of CCK and acetylcholine, the synthesis of zymogen is reduced. So here's just basically a closer look at the secretion process, which we mentioned back here. So zymogen granules, what are these? So these are important specialized organelles in the acinar cells that are key features in the release of degradation enzymes. So some research into these show that they contain 5 functional groups of hydrolytic enzymes and proenzymes, along with amylase, lipase, carboxypeptidase, just to name a few. Some experts have done research into the zymogen granules in terms of pancreatic injury, including tumors. The components of the organelles have been analyzed, including the proteome and proteins in the pancreatic juice. It is shown that there are pancreatic cancer proteins released into the pancreatic juice, giving an easy source of early detection of pancreatitis. So in conclusion, the acinar cells play a vital role in protein synthesis in the pancreas. These enzymes produced are major components in protein degradation, along with other dietary biomolecules, for example lipids and carbohydrates. The enzyme secretion begins in the zymogen granules as they are moved to the cell membrane and into the pancreatic ducts. Neurotransmitter and intestinal hormones such as secretin and CCK activate this process. Zymogen granules are important players in the storage and secretion of the degradation enzymes, along with playing roles in pancreatic injury and disease. So that concludes our take on the acinar cells.